Okay, so today we have Dr. Nikki Farnsworth with us. She's coming from Colorado School of Mines, and her talk is Stressed Beta Cells Contribute to Loss of Peri-islet Extracellular Matrix in Type 1 Diabetes. A little about her. She's an assistant professor of chemical and biological engineering at the Colorado School of Mines, as I mentioned. She's also affiliate member and adjunct assistant professor at the Barbara Davis Center for Diabetes at the Anschutz Medical Campus in Colorado. Prior to joining the Mines facility, she completed her postdoctoral training at Barbara Davis in the bioengineering and pediatrics department under Dr. Richard Benninger, working at the intersection of cell biology, diabetes, and biophysics. She completed her PhD and MS degrees in chemical and biological engineering at UC Boulder and completed her BS in chemical engineering at Rensselaer Polytech. Her work is funded by Breakthrough T1D, the Federation for Diabetes Research, the American Diabetes Association, NIH, NIDK, the NIH Diabetes Research Center, and Helmsley Charitable Trust. And finally, she was a recipient of the George Arjun Barth Award from NPOD. So congratulations on all those accolades. And we're really, we can't wait to hear more about this recent preprint and paper. And I am asking people to put questions in the chat during the talk. She'll answer at the end. And also there's a copy of the recent paper there for your reference. Thanks so much for joining us.
Thank you so much for this opportunity to talk about this work. I'm really excited, and I'm really have to gush a little bit about all of the work that you and the Sugar Science do. I think it's amazing. So I'm really thankful for this opportunity to talk a little bit about our new work. So given the background of everyone who is here to hear about this paper, I probably don't need to tell you what type 1 diabetes is, but I really wanted to kind of step back and refocus on what the distinct stages are and how we classify the stages of type 1. So generally, initially, we have some kind of genetic susceptibility or environmental risk factors for developing type 1, and then we have a triggering event that initiates that autoimmune response. So in stage 1, these folks are typically autoantibody positive for at least one autoantibody. The immune process has started. There may be some infiltration of islets, but these are normoglycemic and asymptomatic individuals. As we progress to stage 2, where we lose a significant amount of beta cell mass, there may be some dysglycemia. It may be asymptomatic or slightly symptomatic, but this is typically where we see multiple autoantibody positivity. And then they decline further into stage 3, where they are hyperglycemic. This is typically where clinical diagnosis occurs and where we have multiple autoantibodies generally. So the factors that lead to this really quick decline in beta cell mass, we don't really understand very well. And a lot of the focus has been on either the immune component or the beta cell component. But I want to draw some attention to the microenvironment that the islet sits in, because I do think this plays a significant role in disease pathogenesis. And I think within the field, there's a growing recognition that we can't take this islet-centric focus in understanding type 1, that we need to broaden our view to understand how type 1 is impacting other factors, other cell types, and the pancreas microenvironment as a whole. So when we think about the islet microenvironment and the niche that it sits in, there is actually, in addition to the different cell types within the islet and the innervation and vasculature within the islet, there's also 2 distinct types of extracellular matrix, or ECM, that exist. The first is the basement membrane that sits directly surrounding the islet. It forms this beautiful little capsule that creates a niche around the islet. This is composed of really unique components, things like laminins, collagen type 4, which is a network collagen that's quite soft, perlecan, and nitrogen. So these components are mostly found around vasculature, but we also see this surrounding the islet as well. Then we have the interstitial matrix, and this is sort of the bulk ECM of the pancreas, where this is in all of the other parts of the pancreas, endocrine or exocrine. This is composed of a bit stiffer components like fibronectin, fibrillar collagens like type 1 and type 3, and then sulfated glycosaminoglycans like hyaluronan, chondroitin sulfate, and heparin sulfate. So each of these components plays a different role in supporting islet function, particularly when we think about the components that are directly surrounding the islet, like perlecan, collagen type 4, and laminin. As we can see in these IHC images, they form this really unique environment that provides cues, both mechanical and biochemical, to the islet that help to support its function and its survival. In type 1 diabetes, it's been fairly well established, and it's been really well characterized by a few labs, primarily Eva Korpos, Marika Bogdani, Thomas White, have all shown that there are these really unique changes in the ECM that occur in type 1, where we see a loss of the extracellular matrix. In this case, these images are showing peri-islet laminin, and that this loss occurs in conjunction with infiltration. So the degree of insulitis generally correlates with the loss of extracellular matrix. And we've shown this in non-pancreas sections on the left-hand side, as well as in human pancreas donors from the NPOD program. And on the right-hand side, we're showing laminin and collagen type 4 loss with immune infiltration marked by CD45 immune cells. So generally, these studies have shown that there's not just islet-specific changes to the ECM. The changes are actually much more broad than that, and that we lose lots of components of the basement membrane as well as the interstitial matrix. So while, yes, the loss of ECM correlates with degree of insulitis in NOD animals and in type 1 individuals with confirmed type 1 diabetes, this isn't the full picture, because the immune cells are only able to secrete a range of ECM degrading enzymes. And we see this broad loss both in the interstitial matrix and basement membrane that, in my mind, tells us that we're not seeing the full picture here, that the immune cell infiltration may not be the only source of ECM degradation and loss during the development of type 1. So this is what we wanted to really explore in this study. And the motivation for why we think it's so important to understand how ECM is lost and at what point it's lost is because we think this plays a role in the pathogenesis of disease. So previous studies have shown that if we provide NOD mice with a heparinase inhibitor, so this inhibits the activity of this ECM degrading enzyme heparinase, that we see a reduction in insulitis. We see a delay in onset of disease in these animals. Specifically, we're preserving the heparin sulfate, which is stained in blue by this Alshan blue staining on the right-hand side, and that is actually leading to protection against inflammation and beta cell death. So this tells us that that loss of an ECM, specifically surrounding the islet, is important for the pathogenesis of disease. So this is why we really want to study when this is happening and understand how this may be impacting beta cell function and beta cell survival in disease. So like I said, we do see that this loss of ECM correlates with insulitis in the NOD mouse and in individuals with type 1. But we think that there's other components to this. And more specifically, there was a recent paper by the Saunders lab that showed when they stressed beta cells with doxycycline, that they actually see this significant upregulation of matrix remodeling enzymes. And those include things like ADAMTS molecules, MMPs, lots of different varieties of these molecules. And each of these matrix degrading enzymes targets typically a very specific ECM component, either laminins, collagens, glycosaminoglycans. But you can see that they're upregulating a host of these molecules. And in my mind, this was really interesting because the way that I've thought about the islet traditionally is that it does not make or maintain the ECM that it sits in. And generally that's actually quite weird, because every other cell in your body will make or maintain or play some role in remodeling its environment. So it sort of makes sense inherently that the beta cell and the islet as a whole would also do this, but maybe to a lesser extent given the energetic demands of insulin production. So this is really at the heart of what we wanted to look at in this study. We wanted to understand at what point are we getting changes in the islet microenvironment, specifically the extracellular matrix, and how is this contributing to disease? But specifically, what are the contributions to loss of ECM, both around the islet and in the pancreas as a whole in type 1 diabetes? So for us to do this, we wanted to think about what potential stressors there are in type 1 that could be contributing to beta cell production of ECM degrading enzymes. And the thing that comes to mind is pro-inflammatory cytokines, because we know these play a really important role in mediating beta cell loss and dysfunction in type 1. So activated T cells and macrophages produce really high levels of a whole host of pro-inflammatory cytokines, including TNF alpha, interleukin-1 beta, and interferon gamma, just to name a few. And we know that these cytokines induce beta cell stress in vitro. This has been shown by my lab and a whole bunch of other labs at this point that cytokines do cause beta cell stress. So cytokine stress in other cell types causes upregulation of tissue degrading proteases. So we wanted to understand if this also happens in the beta cell. So to do this, we started off with mouse islets because this is an easily accessible model for us. And we used a pro-inflammatory cytokine cocktail that in our lab has been shown to cause significant amounts of beta cell stress, but not significant amounts of beta cell death. And so this is a low dose of TNF alpha, interleukin-1 beta, and interferon gamma given over a 72-hour time point. We looked at a broad range of ECM degrading enzymes as a screen initially to look at what enzymes the beta cell might be upregulating. And we found MMP3 to be the sort of standout among the initial screening panel. So we continued with looking at MMP3 gene expression as our marker for upregulation of ECM degrading enzymes in the beta cell. So we found that this low dose of cytokines over 48 to 72 hours in mouse islets significantly increased expression at the gene level of MMP3. And when we treated human islets with the human-derived recombinant pro-inflammatory cytokine cocktail, we saw similar results, that human islets are also capable of upregulating MMP3 under cytokine-induced stress. When we look at the protein level, because the upregulation at the gene level may not necessarily recapitulate what's being produced in terms of protein, we also see increases in protein production of MMP3 at 72 hours in human islets that were treated with cytokines. So this is really encouraging to show that, hey, the beta cell, either mouse beta cells or human beta cells, are capable of upregulating production of these matrix degrading enzymes. And then further, we looked at NOD mouse islets. These are 12 to 15-week-old normoglycemic non-mouse islets that could potentially go on to develop type 1. If we look at the expression of MP3 levels in these NOD islets compared to immunodeficient NOD-SCID rag controls, we do see this upregulation of MP3 in these islets as well. So this shows us that disease-associated conditions in the NOD mouse also stimulate production of ECM degrading enzymes by the beta cell. So we further wanted to look at other types of stressors that you might see in type 1. And the thought process was that we wanted to look kind of across stages, not just at early stages, but also potentially at later stages where we start to get symptomatic individuals. So we wanted to look at the effects of hyperglycemia on production of ECM degrading enzymes. And to do this, we used a high glucose, 20 millimolar glucose, for up to 48 hours to mimic hyperglycemia. And when we treat human islets with this hyperglycemic condition for 48 hours, we see, again, a significant upregulation of MMP3 gene expression, as well as upregulated protein expression of these enzymes. So I think this was really exciting because this was really the first evidence that we've been able to find that the beta cell is capable of remodeling its environment, and specifically the extracellular matrix. So this was really novel for us. So the next thing that we wanted to do was to investigate changes in ECM in the NOD mouse during the progression of disease. And so to do this, we again focused on MMP3 because we saw at the protein level that there was evidence for production of this matrix degrading enzyme. So we used immunohistochemistry of pancreas sections from 12-week-old normoglycemic NOD-SCID or NOD mice. And then we have our hyperglycemic NOD mice that we used as our stressed condition. So in our NOD-SCID mice, we stained for insulin and MMP3 on the left-hand side, and we stain for insulin and collagen type 4 on the right-hand side. These are consecutive sections so that we could try to correlate expression of MMP3 and collagen type 4 in the same pancreas. And we took sections from 6 different depths of each pancreas over 3 animals for each condition. And we eventually quantified these images. But just the preliminary immunohistochemistry, we can see that we have a nice collagen type 4 capsule that exists around the islets in our NOD animals, our NOD-SCID animals, and that generally MMP expression is quite low under normal conditions. As we transition to our 12-week NOD pancreas, these again are normal glycemic animals, but in the NOD mouse, we see infiltration as soon as 4 weeks of age. And so it's not uncommon to see significant amounts of insulitis in a 12-week-old NOD mouse, despite them being normal glycemic. So if we look in the islets that had significant amounts of infiltration, we can actually see that there is an upregulation of MMP3 expression, specifically around the insulin positive areas, but also in the infiltrate. And then if we look at the collagen type 4 staining, we see that we're losing ECM molecules. We're losing collagen type 4, both in the areas that have been infiltrated, but also in the insulin positive areas. So this is giving us some clues that there may be some role for the beta cell in degrading the intra-islet collagen type 4 that we can see here. When we move to our hyperglycemic pancreas, we can see even higher levels of expression of MMP3, specifically in our insulin positive areas. And on the right-hand side, this correlates to an even greater loss of collagen type 4. But what we really noticed here is that while we do see loss of collagen type 4 in the insulin positive areas and in the infiltrate, we also see a loss of collagen type 4 in the bulk pancreas. So we wanted to be able to quantify this to say, what is the distribution? Because we know that infiltration is very heterogeneous. We want to understand how this process is occurring and what the spread of loss of collagen looks like. So to do this, we basically took our IHC images and we used what I'm going to call the semi-custom code that we integrated into ImageJ, which is freely available image analysis software. So this is our IHC image of collagen type 4, and it's overlaid a DAPI channel for the nuclei, insulin staining, and then collagen type 4. And then we use the insulin staining to isolate out only the staining area that was positive for insulin and collagen 4. So we're getting just the islet collagen. And we tried to extend this slightly to include the peri-islet collagen 4 capsule. And then we use this to specifically quantify all of the collagen type 4 positive area within the insulin positive area so that we can look at just the islet specific changes in collagen type 4. So we did this for both collagen and for MMP3 staining. And when we look on the left-hand side, we can see that if we look at our MMP3 positive area versus our insulin positive area, and this is a combination of our MMP3 staining intensity multiplied by the area, so it gives us a semi-quantitative way to look at MMP3 expression. We can see that we have really low expression levels in our islets of non-skid animals. But as we progress to the normal glycemic NOD animals, we can see that there's a slight increase in MMP3 expression. And as we move along to our hyperglycemic NOD animals, we see this significant upregulation of MMP3 activity, which matches what we saw in our representative images. As we look at collagen 4 area in the islet specifically, we see that as we progress to our NOD animals, even though they are normal glycemic, they were all confirmed to have insulitis. And we see a slight decrease in our collagen 4 positive area in those animals. And then as we progress to our hyperglycemic NOD animals, we see this significant decrease in collagen 4 staining area. So to try and stratify this a little bit more and try and understand how this might relate to insulitis, trying to get at a little bit more how much of this is due to infiltration versus beta cell contributions, we were able to stratify our MMP3 staining and our collagen type 4 staining by insulitis score for each islet for each animal. And again, we quantified as many islets as we possibly could find on each section over 6 different depths of each pancreas to try and characterize the heterogeneity of infiltration and of these changes to the ECM across the pancreas. So we can see on the left-hand side that our MMP3 staining for islets that are minimally or not insulitic at all, we don't really see huge changes in MMP3 activity, but as we progress to higher stages of insulitis, a score of 2 or 3, which indicates significant or severe insulitis, we can see that there's a clear increase in our MMP3 activity in our hyperglycemic animals compared to our normal glycemic animals. And again, our NOD skids had almost no MMP3 activity. So this shows us that while we do have MMP3 activity in our normal glycemic animals, it's further enhanced under hyperglycemic stress more significantly. When we look at the collagen 4 staining in contrast, we can see that there is a slight drop in our collagen 4 staining in our hyperglycemic animals that are not infiltrated. But as we progress to higher infiltration states, we see significant decreases in collagen 4 staining. But what's interesting is that if I compare insulated score of 2 and 3, the hyperglycemic animals consistently have greater loss of collagen 4 within the islet. So what this is telling me is that the loss of collagen is independent of infiltration to some degree. Because we have consistently more loss of ECM in the animals that are hyperglycemic, it tells me that the hyperglycemic stress is likely what is driving expression of MMPs by the beta cell, and that this is contributing to loss of the ECM more so than the infiltration. So this is fantastic, and I think this was really exciting for us, but really what we wanted to do was to move into some human samples and show that this is actually happening in human samples, because generally for those that have worked with NOD mouse, you know that the infiltration is quite severe and quite strong compared to what we see in human disease. And so it's really important for us to be able to characterize this in human samples. So to do this, we use samples from NPOD, and I spent a lot of time trying to age and BMI match all of the donors that we picked. And as a preliminary screen, we picked 3 donors from each category, either folks that were healthy controls that had no diabetes. We had some that were recent onset type 1, so less than 6 months after diagnosis that we collected their tissues. And then we had double autoantibody positive individuals that did not have confirmed diabetes. And for the folks that we had where they were confirmed type 1, they were recent onset, they all had an HbA1c greater than 10. So this is our hyperglycemic mimicking condition that we selected. So these were the 3 groups that we analyzed from our NPOD samples. And we analyzed, again, as many islets as we could from multiple sections from each donor. So using the same type of analysis, we stained for MMP3 and collagen type 4 on subsequent sections, and we looked at changes in ECM and changes in MMP3 expression. And something that we noted was that MMP3 expression in the human tissues was naturally much higher across the board, both within the islet and within the exocrine tissue. We see really nice type 4 collagen capsules surrounding the islets and surrounding the vasculature in the human samples, which did mimic what we saw in the NOD samples. When we moved on to our autoantibody positive individuals, we can see that there's an increase in MMP3 expression, both in the exocrine tissue, much more so in the exocrine than we saw in the NOD, but also in the insulin positive areas. And so this really nicely matched what we saw in our sort of normal glycemic infiltrated NOD samples. And then when we look at collagen type 4 expression, we can already see that there is a loss in the peri-islet basement membrane capsule, but also more globally. There's just generally less type 4 collagen in autoantibody positive individuals. When we move on to our recent onset individuals that have a HbA1c greater than 10, we see even more MMP3 expression. But the thing that is unique in the human samples that we did not necessarily see in the NOD samples is that the MMP3 expression is not limited to just the islet. We see it more broadly, that both the exocrine tissue and the endocrine tissue are expressing very high levels of MMP3. And we get this really global loss of collagen type 4 in these recent onset individuals. Some of the things that I will point out, though, it may be a little bit hard to see, but if you look at the paper, we've kind of separated out each of the different channels. In the individuals that are recent onset, we actually see enhancement of collagen deposition around the vasculature, but not a recovery of type 4 collagen around the periphery of the islet or in the exocrine. So this is really interesting to us that we get these sort of different changes in the ECM depending on which compartment we are looking at. So again, we wanted to quantify this information, and so we quantified our MMP3 positive staining area per insulin positive staining area, and then our collagen 4 staining area as well. And we saw that as we progressed from healthy to autoantibody positive, we get this increase in MMP3 expression in our autoantibody positive population, and in our recent onsets, hyperglycemic, as defined by HbA1c values, we see even greater expression of MMP3 specifically in the beta cells. But again, we also did see this in the exocrine tissue as well. And this correlates with a loss of collagen type 4, both in our autoantibody positive and our type 1 diabetes recent onset patients. So overall, this data correlates really nicely with our NOD data and suggests that we are getting some beta cell component of ECM loss in type 1 diabetes, and that we see this more mildly happening in autoantibody positive individuals. And we see this progressing as we start to develop symptomatic type 1, that we're getting more and more contributions of the beta cell to loss of the ECM. So where we think this is sort of fitting into the big picture of type 1 progression is that this may not be an initiating event in type 1, but it could help contribute to the acceleration in beta cell loss that we see. If the beta cells are degrading their environment, they are permitting the immune cells to more easily infiltrate and more easily attack. The ECM is providing really critical cues that help to support the islet function and survival. So when we're losing this, this could make it easier for the beta cells, for the immune system to kill beta cells, and could help contribute to the super sharp decrease in beta cell mass that we see prior to disease onset. So I think this is really interesting information, but we wanted to kind of more broadly look at ECM remodeling capabilities of the beta cell. And so to do this, we were super fortunate that we were able to work with some collaborators who had already published a proteomics data set that they collected from a set of 10 human islet donors. And these are isolated human islets that were either untreated or treated with a cocktail of pro-inflammatory cytokines, interleukin-1 beta and interferon gamma for 24 hours. This data set is freely accessible. This is the GEO accession number if you'd like to also go back in and reanalyze this data set, as we did. But we basically took this data set and reanalyzed it to focus around ECM regulation. So any signaling pathways that have to do with regulation of the extracellular matrix in the beta cells, this is where we focused our analysis. And when we look at a volcano plot of significantly upregulated and downregulated transcripts that are just associated with ECM, we see some really interesting things that, in addition to our MMP3, which you can see kind of highlighted on the bottom, we get upregulation of a host of other ECM degrading enzymes, including ADAMTS1, MMP1, ADAMTS4, MMP10, and MMP25. And each of these targets a very specific ECM component. Most of them, though, are actually focused around collagens and collagen type 4 specifically. So we see that there is some role for the beta cell in remodeling collagen type 4. And when we look at the significantly downregulated transcripts, the interesting thing that we see is that collagen itself, collagen type 4 or collagen profibril formation, is significantly downregulated. So in my mind, this is telling us that the beta cell is not an innocent bystander in mediating its environment. It is actively remodeling that environment. Is it to the same extent that an endothelial cell might do it? I don't think so. And that's because of how energetically demanding insulin production is. So I think what this is showing us is that, to a lesser degree, the beta cell can help to remodel its environment and to mediate the extracellular space through the extracellular matrix. But I don't think the turnover, and I don't think the contribution is as big as some of the other cell types in the pancreas because of that energetic demand of insulin production. So when we look a little bit deeper at the upregulated and downregulated pathways that we were able to pull out through GO gene ontology analysis, we see that we have significantly upregulated ECM organization, proteolysis of the ECM, collagen catabolic processes. So those are some of those MMPs associated with collagen degradation, and then ECM disassembly. And then we get upregulation of, we get downregulation of collagen formation, which again relates to those collagen genes that we were able to pull out, cell adhesion, and ECM organization. So overall, this suggests that inflammation or beta cell stress in general is triggering a transcriptional program that is promoting ECM remodeling. And when this is a chronic inflammation, we can envision how this might lead to a pro-degradation phenotype where we are just losing ECM faster than we're able to help remodel it and bring back that ECM. So overall, what we think we've shown in our study is that stress beta cells do contribute to ECM remodeling by upregulation of MMP3, as well as a host of other ECM degrading enzymes. Loss of ECM and MMP3 expression we saw began in our autoantibody positive individuals, but we think this is exacerbated by stress, and specifically hyperglycemic stress, that can be independent of immune infiltration, and that this may allow the immune cells to actually infiltrate the islet more easily and allow them to more easily kill the beta cells, that leads to this super rapid decline into clinical diagnosis and stage 3 of disease. So this is definitely a very preliminary study, and that there's a lot of questions that we still have. So we want to better characterize changes in other ECM components like laminin and sulfated glycosaminoglycans. We want to also demonstrate more specifically that beta cells are the ones that are degrading that ECM. The evidence that we have, I think, strongly suggests that they can, but it is still highly correlative. And so one of the focuses of my lab is to use biomimetic biomaterials where we can basically recreate that extracellular matrix environment of the pancreas and encapsulate human islets and stress them to see if they can actually degrade that ECM. So those are the next steps for this study. And then we want to also expand our analysis of our NPOD tissues to include sex-based differences in MMP3 expression and ECM loss, because there's a lot of mounting evidence to suggest that there are sex dimorphic responses of the islet to inflammation and inflammatory stress. And I think this may lead to differences in ECM loss that are sex-specific. So this will be a really interesting next step for this study. So I'd like to thank everybody who worked on this study, specifically my newest minted PhD student, Chelsea Johansson. She just defended her PhD about a month ago, and she is wrapping up her studies. And then Kennedy Lamb, who is taking on this study. I really want to thank NPOD for providing us with pilot funding to do this work, and for also giving us access to these tissues, as well as the organ donors who provided these tissues, because it's such a critical resource, and we're so grateful to have these to be able to look at human disease progression. I'd love to take some questions about this. I know this is maybe a little bit long of a talk, but there's so much to dig into here. So thank you all for your
Yeah,
attention.
no, it's well worth the length because it was such a great talk. Yeah, I have a couple questions, but there's already some questions in the chat, and so I'll read them out to you. Here from Sarah Richardson's group, or Sarah Richardson herself is here. They recently published and discovered and published seminal work describing the small endocrine objects. I'm sure she has a question. She says, congratulations to you and the team. Have you combined hyperglycemic stress and cytokines together to see if this effect is additive?
Yeah, that's a great question. We have not done that yet, mainly because I am concerned that we are going to kill the cells, and I want to look at stresses that are not necessarily lethal. But I think that would be a great thing for us to try as well, and we could maybe reduce some of the cytokine load to see how additive that is. But I would anticipate we would see very similar and a very strong response, which might better mimic the type 1 conditions that we are trying to mimic in vitro. That's a great suggestion.
Yeah. And sort of tapping back onto the Richardson group's work, like how might your work, can you expand, like how might your work, what you found, intersect with her novel discovery of the small endocrine objects? Could endocrine object loss be partly, a matrix failure phenomenon? I mean, I don't know, maybe if you want to talk about it together, feel
Yeah.
free to unmute yourself, Sarah, I don't know.
Hi, Nikki. Great talk. Well done. Yeah, I guess, I mean, our thinking is that perhaps the smaller endocrine objects don't have quite so much of the matrix established around them in the first place, which is probably why in individuals who are getting type 1 diabetes, they're the first things picked off by the immune system because they don't have those defenses. But I think this is a beautiful way of illustrating how the larger islets that seem to be able to be preserved post-diagnosis, at least in some individuals for a long period of time, how this gradual destruction and loss might be happening. So I really love the story. Well done.
Oh, thank you. I think it is such an interesting concept because, like the publication that you put out, I was like reading through this as we were doing our analysis, and I was looking for the small endocrine objects and, the exact same conclusion that it's like, okay, they're not there, but I'm keep trying to think about how we can answer the question of what happens to them. And so I think what we might try, we have these these biomaterials that we can functionalize with ECM. And I think it could be interesting to do sort of a like diabetes in a dish type experiment where we have different size of clusters, and then we can treat them with splenocytes from a diabetic NOD just to see what is the difference. Is it that they're being killed off sooner? Is it, something to do with the ECM environment? If we tweak that environment, is it just not permissive for them to continue to be beta cells? So I think there's so many interesting questions, and this is where I would love to loop in someone who has a developmental biology background to understand, is it loss of them? Is it dedifferentiation, transdifferentiation? That piece I have not wrapped my head around. Do you have thoughts on that?
Yeah, I mean, we're working quite a lot on trying to sort of figure out what they are and where they come from, and also trying to do some of the staining for looking for the kind of collagen 4 and things like that around those smaller endocrine objects. So yeah, very happy to kind of hook up and have a discussion maybe separately on here, so other people get a chance to ask some questions, because we could probably take up the rest of the 20 minutes.
That sounds great, like a collaboration brewing. I did have a question for you, Nikki. Do beta cells sort of like undergo cyclic ECM modeling during their development? And do they sort of,
That's a fantastic question.
and how, and like if they are sort of young for their age in some way, right? They're not fully mature. Could this impact that ECM remodeling?
It's such a great question. And I think the thing that always blows my mind is that I don't think we have any evidence to support beta cell ECM remodeling and production. And I think it's partially that we haven't looked. So I think that is a really ripe space where we should be looking at what role does the beta cell play in contributing to that environment, because I think there is a strong evidence to support acinar production of ECM, endothelial production of ECM, but I think we sort of have ignored the beta cell, because I think if we were to categorize it in terms of, what is the percentage contribution to the completed ECM, I think the beta cells are a lesser percent than the acinar and then the endothelial cells. But I think they are still a contributor. So I think that would be a really interesting thing to look at in development. If we eliminate the beta cell's ability to do that,
Gotta
do
get
we
the
see
developmental
significant
person.
changes?
Yeah.
Yeah.
There's so many fun experiments for you guys to do. David Hill's asking or saying, excellent work, Nikki. Do endothelial cells and parasites have a role, or are the vascular cells only involved with inflammatory cell actions on ECM remodeling?
Yeah, it's such a great question. So we do see that there is a role for parasites, and this is work that stems from Joanne Almas's investigations into parasite function and dysfunction and ECM during type 1 diabetes. So she has shown that there is this thickening of the vasculature ECM, and that it's driven by parasite dysfunction due to inflammation. And I think we see some of that as well in our samples. We see very similar results where the vasculature specifically is thickened as you move from autoantibody positive to confirmed type 1. We don't quite understand why that is, or how that's contributing to changes in islet function, but I think it's actually a distinct niche compared to the rest of the ECM, because we don't see that same change in like the peri-islet BCM. And in the majority of samples that we looked at, we actually don't see recovery of that peri-islet collagen 4 capsule, and there's still a global loss of collagen type 4 in the exocrine pancreas. So I think there are distinct niches that are regulated by the cell types that sit in them, like the parasites and endothelial cells. And I think they're completely different problem compared to like the peri-islet BCM. So I think there's different things that are regulating those unique niches. I do think there's crosstalk between them, because I think the interactions between the parasites and endothelial cells and the resident alpha and beta cells are important in regulating beta cell function. But I think we're sort of just discovering what those are and what role they're actually playing.
How, is there, we talked briefly about this offline, but can you just touch on the global ECM abnormalities that, yeah, I mean, you have to have your correlative tissues, right, with your samples. So it's a big ask, but have you seen any evidence of ECM changes across other tissue types in these, say, 1 or 2 autoantibody positive individuals?
So we haven't necessarily looked at other tissue types. Um, I think there was some evidence or some studies, I can't remember how long ago, but it wasn't particularly recent, that tried to think about ECM remodeling as a way to provide a biomarker for development of type 1. And I think it's really tricky because the quantities of ECM that you're seeing are not huge. The cells are really good at trying to kind of clean up the microenvironment. So there's not large amounts of ECM just kind of circulating in the vasculature and in the bloodstream. Yeah, but I think in terms of like the pancreas and all of the different cell components that exist within the pancreas, I think what we've seen, at least in our data, is that the acinar cells are likely also significantly upregulating ECM degradation, and that they can also kind of shift their phenotype from more of a remodeling to just a degradation phenotype as we get this more and more inflammatory assault, and as it goes from a kind of acute to more of a chronic situation. I think we're just seeing this permanent phenotype shift. So I'd be really interested to see what role does the acinar cell play in contributing to more of the global change in ECM, and is that why we see these changes in pancreas mass and pancreas volume that we see? Is that the cause of this? Is there something that we can use to better identify this?
Yeah, no, that's great. That's a great comment. Th1nk, thanks, Nikki. Excellent talk. Regarding ECM loss, beta cell stress. Oh, ACM loss, beta cell stress, and insulitis are so interconnected. Do you have any thoughts on how they contribute to islet heterogeneity within a given donor?
Oh, that's such a great question. I,
Why is it Apache?
yeah, that's a great question. I think I don't have a good answer based upon our data, but mainly because I don't think we have enough data. This would be a fantastic project if I could find a student that is really good at writing code and that we could use machine learning. And like realistically, to answer that question, I think we need a lot of data sets. I think it's doable, but we need to analyze as many patients as possible and try and get some better insight into, is there heterogeneity in ECM? Where do we see it? What does it correlate with? So I think that would be a really fantastic question for someone that has a machine learning background to help us answer and say, okay, can we collect as much data as exists, and can we try and tease out some of these correlations? And then we can go in and say, okay, is there a way that we could either in vitro or in vivo test this correlation and understand that? But I think as it stands with our study, we don't have enough statistical power to say something about it. But it's a really fantastic question, and I would love to see the answer to that.
Well, maybe someone will take it up in our upcoming D challenge and start parsing through some data. And that's what we're asking people to do, is use some of these new AI/ML tools to parse through data, looking for new correlations. Okay, Jordan Wright, beautiful work. Post-translational modification of ECM like glycation likely contributes to diabetic kidney disease. Do ECM post-translational modifications play a role in the islet? Ooh, nice.
That's a fantastic question. And I think the answer is unfortunately maybe slightly disappointing, that no one's looked that I can find. So I think that is another space where modification of ECM, but the other piece of that is, once you degrade ECM, yes, it gets cleared out, but the products that are produced by ECM degradation can also be inflammatory and can also cause dysfunction. In lots of other tissue types, this happens, particularly things like glycosaminoglycans. When they break down into these really small bits that can actually be taken up by cells. They have receptors for these small bits, and that can cause in and of itself a signaling cascade that can lead to enhancing inflammatory responses and leading to more and more dysfunction. So I think both of those components are really, really interesting. I don't think I've seen any evidence for that in type 1, but I wouldn't doubt that it exists. It's just a matter of, can we look for it?
Great, good answer. We're really excited to see what potential collaborations maybe come from this talk, because this is a very fertile area for inquiry, and You've got what you're doing combined with what the Richardson Lab is doing at Exeter, two big powerhouses. So hopefully you guys will join forces and get to the heart of this issue. Thank you. Have a great rest of your
Thank you.
day. Bye-bye.